All right, good afternoon, everybody. So I'm so very excited to introduce our next speaker, Greg Miller. My students in the lab have probably heard me say 30 or 40 or 50 times that if I had to do grad school over again and work in an area of research that really excited me now, I'd really want to learn about the topics that Greg will cover today. I think it's fascinating, the intersections between immunological and regulatory functioning and mental health. The work is so elegant, so exquisite. And so I already had a really deep appreciation for Greg Miller's work. And as a consequence of all of this, of course, being the research dad that I am, I always try to live vicariously through my kids, and I always tell them, look, my dream is to place one of you in Greg Miller's lab so you can tell me what it's all about. I got the next best thing. My close friend Katie got postdoctoral training working with Greg, so I had some idea about what it was like. And I already really admired Greg's work, and then I got a chance to see what kind of mentor he is. Yeah. Everyone, I give you Greg Miller.
Thanks, Andy. It's hard to go forward after an introduction like that. It was the nicest introduction I've ever gotten, and I'm not kidding. Thank you. I'm going to move that away so I don't spill Coke all over somebody's laptop before I get started. So thanks for having me here today. Thanks for all of you for coming out on this Saturday. I'm happy to make this interactive. So if you have questions or comments, don't hesitate to jump in. We can talk as much as you want. Probably more interesting for everybody if it's back and forth. So I'll just go ahead and get started. I'm gonna, I think, shift gears a little bit from the topics you focused on for most of today and discuss work we've been doing over the years on early life stress, particularly around socioeconomic status, and how it relates to physical health across the lifespan. So you probably all know that in the United States there are dramatic differences in health and longevity that pattern strongly according to race and class. This is a paper that Paula Braveman published a few years ago summarizing some CDC data. And what you can see is that if you operationally define socioeconomic status according to percentage of the federal poverty line in terms of household income, you see these linear differences in life expectancy according to SES, and you see it across racial and ethnic groups. So within African Americans, within white Americans, and within Latino Americans, they're all strong income gradients in life expectancy. And I can show you this for a number of different specific causes of mortality, various cancers, heart disease, stroke, or for morbidity. The patterns are generally the same. There are strong income-based differences, strong education-based differences, and strong race and ethnicity-based differences. And these, for this audience, I think it's particularly important to emphasize that these disparities start very early in life. So here you're looking at data on maternal education and neonatal mortality and infant mortality. And you can see those linear gradients I showed you in life expectancy post age 25 are actually present from the very earliest days of life. And these disparities, not only do they start in early childhood, but they continue through childhood and adolescence. So on my right in red, you see differences in obesity, defined as being at the 95th percentile, sex and age adjusted for children. And on the left, you see the amount of carotid atherosclerosis. So you can take an ultrasound of the carotid artery and measure the amount of plaque that's present there, or the thickness of the vessel wall, and it's a good indicator of how much atherosclerosis is building up long before you're going to have a heart attack or a stroke. It's the accumulation of early plaque. And you can see at ages 11 to 12, there's this nice study of a few thousand people showing that according to family socioeconomic status, a composite of different educational income measures, there's a gradient in plaque. Again, it's the same linear effect I've been showing you. So already by early adolescence, you're seeing differences in plaque presence. according to SES. Look at BMI. I could show you slides on diabetes or upper respiratory infection, and they would look fairly similar. And this stretches across the life course. So now we're not going to ask about what socioeconomic conditions relate to in terms of children's health, but we're going to ask about what those socioeconomic conditions in childhood relate to in terms of people's long-term health. So this is a fascinating study published a little over a decade ago now that focuses on a cohort of men who graduated from Johns Hopkins Medical School in the 50s and 60s. That was a time where Hopkins only took male medical students. And they were followed, and they're still being followed as I understand it. And the end point here is the presence or absence of coronary heart disease when the men hit their mid-70s. And what it's stratified by here is a very crude metric of childhood SES, whether the physician's father had a manual versus non-manual occupation when the physician was a kid. And as you can see, there's a difference in the rate of coronary heart disease that favors those who came from childhood socioeconomic conditions that were more advantaged, a dad with a non-manual occupation. And these effects are present, interestingly, in a group that is high socioeconomic status themselves by any definition, right? Remember, all of the subjects here are Hopkins Medical School graduates. If you look at occupational prestige, if you look at education, if you look at income, if you look at wealth, by any metric, these are high SES men. So what these findings suggest, and there are a number of others that reach similar conclusions, was that even with a pretty sizable increase in economic mobility from your childhood to your adulthood, these gaps remain, or some of these gaps remain. And early experience seems to have some lasting imprint that sticks around and affects your health later on in life, and isn't completely erased by upward socioeconomic mobility or better circumstances. That's not to say early experience is destiny and there's nothing that can be done. I don't want to leave you with that conclusion. It's not true. There's definitely things we can do to mitigate the impact of negative early experiences, and those things come in the form of different policies and different interventions. But there still does seem to be, even in the most favorable upward mobility conditions, some evidence of early experience lingering. And then there's next generational evidence that's starting to emerge. The strongest evidence for this in terms of cause and effect definitely comes from animal models, but there's even some suggestive evidence in humans where it's harder to do exactly the right study, but the findings are consistent with the animal data, which suggests causality. These are some data on kids with asthma that we've been studying over the past 5 years. Katie was very involved with this work. So this is 300 or so kids with asthma between the ages of about 8 and 17 that we've been studying in Chicago. They all are receiving medical care in the same couple of clinics for asthma. And we wanted to look at whether the socioeconomic status of the children's parents as children. So we wanted to look at these cross-generational effects was related to how the kid's asthma was doing. And what we see is that if you're looking at a teenager today, the socioeconomic status of his or her mother can tell you something about how the kid's asthma is doing, independent of the family's current socioeconomic status. And so what I'm showing you here is the parent and child's report of the extent of asthma control on my right, and on my left I'm showing you some immunologic measures where we take blood from the child with asthma, and in a test tube we expose it to things that cause the immune cells to get activated and produce molecules that eventually are what causes their lungs to fill with mucus and become obstructed, and for them to experience the clinical symptoms of asthma. So on both a more clinical measure and on this biological measure, we see the same pattern, which is independent of the family's current SES, what happened to the kid's parents in their childhood tells you something about disease. And we've got other data, as do other groups, looking at other childhood health and perinatal outcomes, which suggest the same conclusions. So I guess what I would say here is that there's quite solid evidence epidemiologically, substantiated by stronger studies in animal models, to suggest that early life conditions, particularly around deprivation and also, as Kate talked about, around threat, relate to health across the life course. That's not to say that every disease we care about patterns perfectly according to socioeconomic status, or that the only thing that matters is socioeconomic status. There are other things that clearly matter for health, some of which are completely independent of socioeconomic status, like the genetics people get in the lottery, But there are a lot of things that do pattern by socioeconomic status, like lifestyle in some cases and access to resources, and structural and environmental factors that do combine to create these disparities in physical health outcomes. As I said, the strongest effects are probably in the cardiometabolic domain, if you look at things like diabetes, heart disease, stroke, and also in lung disease, childhood lung diseases like asthma and adult lung diseases like chronic obstructive pulmonary disease. The effects are not as strong or consistent in things like cancer, for example. So when people see data like this, when I give presentations or write papers, there's a common response, which is, okay, these are interesting associations, but it's really not that complicated of a phenomenon. It's all about, and then there are three common it's all abouts I get. So the first thing I get, this is all just a genetic story. And the version of this goes something like, well, there's this basket of genes that predispose people to be unhealthy and kind of stupid and make not a lot of money, and those genes travel in families. And what you're seeing here is basically just a genetic effect, that there's a group of people who just aren't so smart or aren't so healthy, and they have a lot of kids, and you've heard this narrative before, I'm guessing. And I think that I'm being a little bit flip about this explanation, but more often than you'd believe, this is what you get, particularly when you give talks like this to medical audiences. But if we took this explanation seriously, which I think we have to, what would you say about the evidence? And I would say, well, how do you address it? Well, first you can look at studies that have been done in administrative fashion in Northern Europe, where they've been able to match up tax records with health records. And with those sorts of data, you can ask questions like, if a baby is adopted from a low-income family into a high-income family, how does their long-term health look? Are they more or less likely to have a heart attack by the time they're 65 than their sibling who wasn't adopted? And the answer to that sort of question when you look at administrative data is that both the genetics of the parents or both the socioeconomic status of the biological parents and of the adoptive parents matters, right? They have independent effects of about equal magnitude. So that's not to say there's no genetic effect, but it's also to say there's a clear environmental signal there above and beyond the genetic effect in the adoption studies. Now you might argue, and I think there's room for this argument, that those adoption studies aren't perfect. They're a good natural-ish sort of experiment, but without random assignment, you can't be sure. So if you're going to require random assignment as evidence, then I think you look to the animal models. And I think there's now very persuasive evidence in mice, in rats, and in monkeys that if you create early life conditions that resemble as best they can the kind of deprivation and the kind of threat that often accompanies being a low-income kid in urban America today, that there are causal effects of those early experiences on conditions, particularly, as I said, in the cardiometabolic domain, how people regulate glucose, their risk for diabetes, heart disease, long-term health outcomes around that. So this isn't just about bad genes. Genes may have a role, and when I say genes in this context, I'm talking about genes in terms of their function of transmitting hereditary influences. But the environment still has a role above and beyond that. Some people think this is all about healthcare, right? So that's another common reaction to these findings, particularly in medical circles, which is we've got a system in the United States where there's unequal access to care, or even if there's not unequal access, there's unequal access to high quality care. Certainly, differences in care access and care utilization are part of the problem, but most of the disparities in physical health by socioeconomic status are things that start before people even get to the doctor, right? And they're things that we don't have effective preventive treatments for. So it can't just be an access to care thing, right? There are differences in health by socioeconomic status in how people's disease progresses once they get diagnosed. Those are there. But the biggest differences are in incidence in the first place. Even if we had a universal access healthcare system, like you might see in Canada or in Western European countries, you still wouldn't get rid of these disparities. And that's shown because you see disparities like this in countries with universal access to healthcare. So it's not just that. And then there's the lifestyle argument that, okay, we get it, it's not just genetics, there's an environmental effect, but the environmental effect is really about low-income families behaving badly. That's sort of the argument. They're more likely to smoke, they eat, potato chips for dinner, you hear these sorts of stereotypes and narratives. Again, I would say, yeah, there are differences in health behaviors that are part of the story here, but we shouldn't view lifestyle and behavior in a vacuum. A lot of the differences in lifestyle can be fairly easily explained by differences in policy, by differences in structure, by differences in environment that really do constrain what appear to be people's choices and back them into particular lifestyles that might relate to worse disease outcomes. And that even when we carefully measure physical activity, diet, and cigarette smoking, they explain a bit of that socioeconomic gradient, but they certainly don't explain it at all. So there's more to it than that. So if it's not just heredity, and if it's not access to care, and if it's not lifestyle, what is it? How is it that early life socioeconomic conditions and the exposures that accompany them seem to relate to physical health across the life course in the domains that I've been discussing? And this is a question that our research group has been focusing on for some time. And what we try to do is use observational studies and, when we can, experiments in animal models to try and understand the biological mechanisms involved and the behavioral mechanisms, to try to identify protective and vulnerability factors that can either mitigate or amplify the associations between the relevant exposures and the relevant health outcomes, and then use what we learn from those studies to make and inform better policy and better practice. And I'm going to tell you a little bit about each of these lines of work as we go through today. So let's start with the mechanistic work. And let me begin by saying that our focus has, for a long time, in terms of mechanisms, involved thinking about the immune system as a key interface between the social environment and physical health. And we've been particularly interested in the parts of the immune system that are involved in generating and maintaining inflammation. And in particular, this process called non-resolving low-grade inflammation, which means that you've got this chronic, ongoing, low-level inflammatory response that's kind of diffusely spread across your body, usually isn't severe enough that you notice it symptomatically, but it's that long-term inflammation that has become increasingly known as a contributor to lots of different chronic health problems that pattern by SES and that cause a good deal of suffering and disability. And you can see from this slide, I don't know if I've got a pointer here. Nope. Probably going to turn the darn thing off. No. All right. You can see up here, non-resolving inflammation is implicated in the pathophysiology of things ranging from atherosclerosis and obesity and diabetes, those kind of standard cardiometabolic conditions I talked about, but also to lung diseases, COPD and asthma, and to some autoimmune diseases. It seems to be a contributor in a more or less strong way to most of the chronic diseases that we know patterned by SES. And our thinking has, probably can be summarized most easily in this very oversimplified graphic, which is that there's a particular set of cells in the immune system that underlie a lot of the chronic low-grade inflammation. And in their immature form, they're called monocytes, and we're going to study them a lot today. And as they get more mature and migrate out of the bloodstream into tissues in your body, they become macrophages or dendritic cells. And our hypothesis has been that kids who grow up in more disadvantaged environments have monocytes and macrophages and dendritic cells that assume this phenotype that leads to more chronic low-grade inflammation over time. And that phenotype arises because these cells, in essence, turn out to be more reactive to threat state. So what are the threats you encounter if you're an immune cell? I get mobile. That's great. Thanks, Andy. So if you're a monocyte out in blood, or you're a macrophage or dendritic cell out in tissue, what are the threats you see? You see bacteria, you see viruses. You also see your own body's cells that have been damaged through some kind of injury or trauma and are sending out these signals saying, hey, there's a mess here, come clean it up. And our argument has been that early adversity primes those cells to respond to those microbial threats and those injury threats in a more aggressive fashion. And we'll talk a little bit more about what that means in a few slides. And that simultaneously, besides being primed to mount a more strong response to those threats, that the cells of kids growing up in disadvantaged circumstances will be less able to shut that response down. One of the key ways you shut those inflammatory responses down is by making cortisol. Cortisol generally has anti-inflammatory effects. And as I'll show you, one of the things we see is that disadvantaged kids' immune cells don't respond to cortisol as strongly as they should. And that we argue that as a consequence of being more reactive and less able to recover, disadvantaged kids have more of this chronic, ongoing inflammation. And that's what contributes across the life course in concert with other exposures to some of the excess risk for physical health problems. So you might say, okay, how do you get from socioeconomic status to this inflammatory phenotype you're talking about? Socioeconomic status, I can already hear Bronfenbrenner and everybody in the room who's read Bronfenbrenner saying, poverty doesn't cause bad outcomes. It's not poverty. It's all the other things downstream of poverty. Okay, I've heard that argument before. I think the epidemiology would speak differently about that, but it is clear, the point is well taken, that it's not just material hardship that relates to a lot of these adverse health outcomes, that there are lots of exposures at the individual level, at the family level, and at the community level that co-vary with socioeconomic status, particularly in the United States, and those more proximal exposures are what affect the developing brain and body. That point is well taken. So we articulated in a Psych Bull paper about a decade ago what we thought some of these likely pathways were, and none of this will surprise you. You know, we argued, as have others, Gary Evans has done a lot of great work in this area, about the social and physical environments of low-income American kids being different than the social and physical environments of their more advantaged peers. And those differences play out in terms of the availability of caregivers, the stability of families, the level of orderliness and chaos and crowding in households, these kids' exposure to conflict and violence in the home, in the neighborhood, in their schools, and differences in diet and infection and pollution. And again, this isn't to say, far from it, that every low-income kid in the country has all of these exposures. The thing about SES is that it captures dozens and dozens of exposures that are higher probability if you're a low-income kid. But there's no certainty, there's no one-to-one relationship. There are plenty of exceptions. So it's a difficult and an easy thing to study at the same time because the circumstances of every low SES child in America are somewhat different, and you can't make these strong claims. But what we've argued, and I think some of the evidence over the past decade suggests, is that those exposures in turn shape the way kids' stress hormone systems get organized and shape the way their brains develop. And based on that sort of thinking, we've argued that there are two kind of overarching pathways that connect socioeconomic status with this pro-inflammatory phenotype. One of them is kind of a classic David Barker-esque programming or conditioning effect, where early experience gets inside the cells of the immune system and kind of is sticky in a real durable way there. And that's a very classic, as I said, Barker-esque hypothesis that comes out of the animal literature. And that's what people talk about as embedding or programming. But we also argue that there's something secondary going on that's perhaps equally important, which you all know a lot about, which is that our early experiences profoundly shape the way our brains develop and the people we become, and how we engage our social worlds, and how we manage stressors, and how we cope with problems in our lives, and what sorts of ways we
we engage in health behaviors. And our argument has been that those pathways are equally important because over the life course, they continue to affect the immune system. The immune system is not like the way many of us have historically conceived the brain, where everything is set by the time you're 25 or so. The immune system has to keep evolving and keep learning. So does the brain, obviously. But the immune system in particular is always got to be prepared to deal with new threats. That means that there's always going to be some plasticity, and that your behavior and your emotion and your cognition across the life course is going to continue to be important for the way your immune system functions. So we've made the case that both these programming effects and what we call accentuating effects are likely to contribute to this pro-inflammatory phenotype. Although it's a good lesson, whenever people cite this paper, they talk about these effects. Like 90% of the papers that cite it. So you never know what your readers are going to do with what you say. It's a good lesson in writing papers. So let's talk a little bit about what inflammation is at the molecular level, just to get situated before we move into data. So this cell right here is a dendritic cell. So this is one of the things that monocytes transform into when they get into tissue. And this dendritic cell has encountered bacteria. You can see gram-negative bacteria up there, the thing with the tail. And bacteria of this species have a molecule on their surface called LPS. It's that red dot there, lipopolysaccharide. You'll hear more about that in a few slides. And what happens is when the dendritic cell or the monocyte binds that LPS molecule, it sets off a signaling cascade. And what that signaling cascade does, I'm not going to go through all the molecular details here. What that signaling cascade does is ultimately converge on this molecule right here called NF-kappa B, nuclear factor kappa B. This is what's called the transcription factor. And it's the linchpin for the inflammatory response. And what I mean by that is that when this gets activated, it moves into the cell's nucleus, and it binds to the DNA, and it switches on the genes that allow the cell to make the proteins that are involved in inflammation. And those proteins involved in inflammation you may have heard of. They're things like interleukin-6, interleukin-1, tumor necrosis factor alpha. These are just signaling molecules that immune cells send to each other and communicate. There's bacteria in this person's big toe. Get over to the big toe. Kill that bacteria. Okay, this person's fallen and scraped their arm. There's a wound in their arm. Get to the arm and start sewing up that tissue, right? These molecules are just instruction signals that immune cells send to each other, and we measure them in lots of studies as a way to kind of eavesdrop on what immune cells are saying to one another. And so what we're going to do in a lot of studies is we're going to take blood cells from kids and adults, and we're going to put them in a test tube, and we're going to add LPS, this bacterial molecule, and we're going to watch this process happen in a test tube and see how much of these molecules do those cells make, how much NF-kappa B is activated. And that's going to help us understand these hypotheses about early socioeconomic adversity in the pro-inflammatory phenotype. Now, how does this process get slowed down? I talked a little bit a few slides ago about cortisol. So you all know cortisol as a stress hormone. You've probably studied it extensively. It is a major regulator of the immune response. And in fact, when you go to the pharmacy because you have bad allergies or a bad rash or an autoimmune disease, often what you're going to get prescribed is a topical or an oral version of cortisol that's been modified slightly. So prednisone, dexamethasone, those are all just synthetic versions of cortisol. And the reason you take them when you have a rash or asthma or an autoimmune disease is because what cortisol does is fly across the cell membrane and bind inside the cell to its receptor. Receptor is called the glucocorticoid receptor. And the reason that cortisol, or that synthetic version of cortisol, will treat your rash or make your asthma subside is because that complex, cortisol plus the glucocorticoid receptor, tethers nuclear factor kappa B and prevents it from going into the nucleus to switch on gene expression. So it basically ties up this linchpin of inflammation. So we're going to study that process too. All right, let's get to some data. Does anybody have questions before we move into some data? All right, so this is some of our earliest work in this area. We wanted to simply ask this question in about 2007 when we started noticing this literature on early socioeconomic status and later health, of whether the data were consistent with a scenario where the socioeconomic status that people had in childhood could tell us something about their immune system's functions when they were adults. And in particular, whether this inflammatory phenotype that we hypothesized might be present. So we did a very simple study where we recruited young adults between the ages of 20 and 45, and we recruited half of them to have come from families of low socioeconomic status, meaning that during their first 5 years of life, their parents had occupations that were mainly in the service sector and the clerical sector. And we recruited a comparison group of young adults who were matched on age and race and ethnicity and gender, who came from more privileged families where during their first 5 years of life, their parents were physicians, were attorneys, were judges, were architects. But we matched on current socioeconomic status. So we made sure that there were no differences between the groups on their current occupational prestige. And in doing so, tried to our best to isolate the effects of early SES rather than later SES. So we gathered this group of subjects, we took blood, and as I showed you in that last cartoon, in a test tube, we treated people's immune cells with various microbial products that made the cells think that they were inside the body and seeing a microbial threat. And I'm showing you here two graphs of different microbial threats that we used in test tubes in that experience. Over here on this side is poly-IC, which is a synthetic substance that mimics a certain kind of double-stranded RNA virus. That's a common virus you experience. On the other side is flagellin, which is a molecule that sits on the tails of bacteria and allows them to move around. And what you can see here is that for both this viral stimulus and this bacterial stimulus, the young adults who came from more disadvantaged circumstances in early life produced more of the inflammatory mediator, right? Now, you might ask, isn't that adaptive? Don't you actually want your immune cells pushing out more of this communication molecule that's going to immobilize the threat and get you back to a healthy state? And that's a good question. And I would say probably the answer in that moment is yes, right? That you want a stronger immune response if your spouse or your schoolmate sneezes on you and is spreading a bunch of influenza virus. You want to get rid of it. So we wouldn't necessarily argue that this is maladaptive in any way. I think what we would argue is that there's a trade-off in that every time your immune system sees a threat, it has a bigger response. That trait might be beneficial in early life, and it might be beneficial in the context of infectious disease. But since this chronic low-level inflammation is the culprit in some chronic diseases of aging, and particularly heart disease, that the trade-off is that you're going to be more at risk down the line for those sorts of diseases of aging. So it's not just a deficit story or an exaggerated response story. It's probably more of a trade-off to particular ecological conditions. So in this study, we did a couple of other things. So one is that we were very interested in trying to not only listen in on these signaling molecules that immune cells use to communicate with one another, but to listen in on the genome and understand which genes were getting switched on and which genes were getting switched off as a way to tell us a little bit more about what was happening inside the cell and understand that machinery. So this is just a summary of what we find in some more complex bioinformatic analyses. The punchline here is that even when people's cells were at rest, this is not after any kind of stimulation with a microbial threat in a test tube, just at rest. What we see is that the subjects who came from families that were more disadvantaged had more activation of this nuclear factor kappa B pathway. And you remember perhaps from a few slides ago, nuclear factor kappa B is that linchpin of the inflammatory response. That's the transcription factor that goes in and switches on all the genes involved in coding for other inflammatory proteins. So even at rest, these cells seem to be primed. They seem to be ready to go. And they have less activity of genes that involve that glucocorticoid receptor. Basically, what this slide is showing is that the glucocorticoid receptor isn't as active at keeping a lid on the inflammatory activity of these cells in subjects who come from more disadvantaged backgrounds. If you want to use a sort of car metaphor, what data like this kind of suggest is that people who come from more disadvantaged circumstances in adulthood, their cells have their feet slightly more on the gas and slightly less on the brakes of inflammation. Right? The cells are just primed and ready to go. All right. So since all of you are developmentalists, you might be asking, okay, so there's at least 25 or 30 years between the exposure to low SES and the process you're studying in adulthood. What happens in between? That's what really matters. So I'm not a developmental psychologist or a child psychologist by training. You probably already figured that out. I'm a health psychologist. But I was fortunate to have Katie as a postdoc, and Katie, along with several of your colleagues, told me that to really understand this and study this, we needed to move back and do developmental research, and that was good advice. So all of a sudden, after spending 20 years studying adults, I started studying kids, and we're now finally done with, I think, a good-sized, decent study of kids in Chicago. This is about, I'm going to show you data from about 300 kids who we recruited in 8th grade, and we followed again in 10th grade. So we've got now 2 years of longitudinal data. And basically, we see pretty similar results. That if you look at 12 and 13-year-olds, according to socioeconomic status, the kids who are living in families that are more disadvantaged have way more activity along this NF-kappa B axis. Again, this is the linchpin of the inflammatory response. And you see this in those monocytes that we've been focusing on. So we pull them specifically out of blood and kind of fish out the cells we're most interested in. You see that pattern. And we also in the study went a little bit further. Not only did we fish out the monocytes, but we fished out the stem and progenitor cells. So basically the mother cells that differentiate ultimately into a bunch of different immune cells. And we show the same pattern in those cells. And the reason that's important is because your monocytes and many of your immune cells die in periods of weeks to months. They turn over very quickly in the body. So if you're going to have any kind of programming effect along the lines that Barker talks about, where experience is sticky and it hangs around in cells, it has to hang around in these stem and progenitor cells because they're the ones that continually repopulate tissue. So these findings give us some hints that that might be going on. So we see these NF-kappa B effects. Interestingly, in kids, we don't see any differences along this glucocorticoid receptor axis. So the insensitivity to glucocorticoids that we see in adults related to early experience isn't visible at 12 years old in otherwise healthy kids. It shows up later. But that's not inconsistent at all with the idea that your experiences can accumulate over time. And we do see some evidence of that glucocorticoid insensitivity in kids with health problems, and particularly in kids with asthma. But in healthy kids, it's more just the foot on the gas difference that we see. So moving back further in development, Katie got me on this trajectory of studying kids, and we actually, over the past 3 or 4 years, have moved even further back, and we're doing some studies in pregnancy and trying to ask, when does this inflammatory process begin? Does it begin in utero? So you guys are all probably familiar with the placenta, right? This is the interface between the mom and the fetus. And what you see here in this nice diagram of the placenta is that it has a key part is this villus layer here, right? This chorionic villus layer. And this villus layer is where exchange between the mother and the fetus occurs. So maternal blood comes in from this side, and it pours into this area through these spiral arteries that develop. And in that blood is nutrients and is oxygen. And nutrients and oxygen diffuse across these villus trees here that are fetal in origin and then make their way through the umbilical cord to the fetus. And what comes out on the other side is that fetal waste comes out here into the maternal blood. And so that's the site where you really have this interface between the mom and the baby that's critical to all sorts of development. And it turns out that in some pregnancies, you can get pretty severe inflammation in this villus layer. And what that inflammation does is obstruct the transfer of nutrients and oxygen. And so this slide right here shows you on a microscope the upper section, A, labeled A, is a cross-section of a normal villus tree. And you can see all these red dots in there. And these red dots contained within the larger purple-pink areas are fetal capillaries, right? They're basically these arms of the tree where maternal nutrients and oxygen can diffuse in and get to the fetus. What you see, so that's a normal cross-section of the villus tree, of a villus tree. In B here, we have one that's chronically inflamed. And particularly right here, you probably can't see it, you have the infiltration, all these purple dots signal infiltration of predominantly maternal immune cells. And there are no red dots here. All of the fetal capillaries have been obliterated by virtue of the inflammation from maternal cells. And so, and it's showing you in adjacent villi, in this case, there's plenty of fetal capillaries. You don't have inflammation, that it's pretty focal to this one. So in our first pregnancy studies, we just wanted to ask, are there differences in inflammation in this villus layer according to maternal socioeconomic status? And so we started doing some work with a placental pathologist, and in a very straightforward, simple study, the answer we got was yes, and they're pretty linear and pretty profound. What you're looking at here on the y-axis is just, is there any chronic inflammatory lesion in the placenta at the time a woman gives birth? And we're looking at lesions in the villus area, but also in this particular study, we're also looking at lesions on the maternal side of the placenta and in the membranes. And in all three areas, there are differences by socioeconomic status. This is an aggregate measure. But you can see that a woman from a low-income background is twice as likely to have a chronic inflammatory lesion in this study as a woman from a high-income background. Now, I should say that most of the lesions we see in this work are fairly low grade. Pathologists typically rate them low, medium, high grade. We don't see a ton of high-grade lesions, and those are the ones where clinically one would be very concerned about risk for fetal growth restriction and adverse neurological outcomes in the long term. These are kind of mild lesions typically, but they do pattern in the way we'd expect. We've also gone, and our postdocs and graduate students love this, we actually show up in the delivery room, and once the placenta is delivered, we biopsy parts of it. So we actually cut it open, and we take out little rice-sized pieces of that villus layer. And then we can use molecular techniques to study the gene expression patterns and the DNA methylation patterns in that place where the maternal and fetal communication occurs. And so we can kind of dissect that molecularly. And when we do that, this is our first study in the area. And it's, it's a small kind of proof of concept study. So I want us to replicate it in a larger sample before I get too confident. But the pattern we see in these little biopsies we take is that the lower income women have more evidence of immune activations and kind of low-level inflammation in the villus layer of their placenta. And the placenta also sends out a bunch of signals that encourage fetal development. So the placenta sends out signals that cause the brain to keep growing, that cause the heart to keep growing. And what we see is that in lower income women, these signals are kind of turned down relative to higher income women. Again, we don't know how clinically significant, if at all, they are. We don't have a big enough sample in this study to really look at developmental outcomes. And even if we did, I'm not sure differences of this size would merit any clinical concern. So I don't want to overinterpret the findings, but what these preliminary data do suggest is that there's a sign, even in utero, of socioeconomic differences in what's happening in that key placental layer. And so we're following this up. We're about 200 women into about a 700-woman study where we're going to have a relatively large sample for this kind of biopsy genomics work to re-ask these questions and see how things look and be able to relate it to more developmental outcomes. So I've talked all about risk up until now, right? I have completely overlooked the fact that we and everybody else in the room knows that the effects of socioeconomic status and other early adversities are usually highly conditional on other characteristics of kids, of the families they live in, of the communities they reside in, and that protective and vulnerability factors are very, very critical to not only acknowledge, but to study. So let me tell you a little bit about our work in this area. So the first thing we found, and I'm going to kind of blitz through this so I don't go too long, is not going to surprise any of you, which is that we consistently find that for low-income kids, having supportive parenting, and particularly a nurturing, caregiving environment from the mother, is quite protective and almost completely eliminates the effects of socioeconomic status that we see on most health outcomes. And we see this when we look at pro-inflammatory activity in cells. So basically having high maternal nurturance obliterates most of the pro-inflammatory effects of low SES. We see this in larger cohort studies. These are data from MIDAS, where we're looking at childhood SES as it relates to risk for metabolic syndrome in midlife. And what we see in that case is that there is a socioeconomic gradient in metabolic syndrome. If you grew up in a household of lower socioeconomic status, you're more likely to have metabolic syndrome at midlife, metabolic syndrome being this constellation of risk factors for heart disease that include abnormal cholesterol, abdominal obesity, and high blood pressure, and fasting glucose impairments. But that gradient is almost completely absent if you simultaneously recollect having high levels of maternal nurturance. And we see this pattern over and over and over again in different observational studies at different stages of the life course. Again, to folks who study developmental psychopathology for a living, this will not be a surprise. This will be very familiar. But to folks in the physical health arena who haven't done this work, this was surprising and reassuring about an important protective factor and one that's highly modifiable. Along those lines, a few years ago, we began collaborating with one of Katie's colleagues at Georgia named Gene Brody, who's a fantastic developmental psychologist that spent his career studying rural African Americans around Athens, Georgia, and has pioneered family-oriented interventions that are administered sometime around the transition into middle school and have strong and durable effects on children's mental health, academic outcomes, and a variety of different human capital outcomes. But Gene historically hadn't been interested in physical health, and that's where we came in as collaborators. And so one of the first things we did is go out into the field and collect blood samples from a few hundred 19-year-olds who, when they were 11 years old, had been part of Gene's randomized clinical trial testing this intervention. So that intervention wasn't designed to look at physical health. It wasn't designed to look at inflammation. The goal was to reduce drug use and to improve mental health outcomes. But he was still following them 8 years later, and we said, why not take a look? And what we found was that the intervention was associated with reduced inflammation 8 years later. Just to give you a feel for the sample, because these aren't the usual samples you see in psychology journals. So again, these are rural African Americans from a few hours radius around Athens, Georgia. About, what am I looking at here? About half the sample has an income to poverty ratio below 2. The percent with an AA or a BA degree in the sample is about 10%. So most of the families have a high school education or some college, and the modal family is a single caregiver household. So it's a demographically at-risk sample. And what we see is that when you look at age 19, at kids who got the intervention versus kids who don't, that there are marked differences in inflammatory markers. And this is a composite of 6 different measures of inflammation that we use. The D values here on individual measures range from about 0.4 to about 0.6. They're higher than I would have expected. But you can see they're whoppingly different here 8 years after the intervention. Now you might say, what happened? How is it that an intervention at 8 years old, or at 11 years old, changed inflammation 9 years later? We did a bunch of analyses trying to understand what roles different sort of psychosocial and familial changes played. And the big operator here was differences in parenting, which is what the intervention intended to do. The goal of the intervention was to increase nurturant-involved parenting and to decrease harsh, inconsistent parenting. And in families where that happened and you saw big changes in overall parenting by latent difference scores, you showed the biggest reductions in inflammation 8 years later. So we don't know, because the study wasn't designed to measure inflammation, exactly what the critical ingredients for changing that was, but we are pretty certain that it has to do with changes in the parent-child relationship that played out over the course of early and middle adolescence, and we think played a role in helping children manage some of the challenges that came with moving into middle and later adolescence, around coping with racism and discrimination, around coping with limited opportunities in these rural small towns, and in coping with physical activity challenges in small towns that don't necessarily have easy places to exercise, and in healthy diets. Those are sort of our hypotheses, and we're going into the field hopefully in about a year to try to replicate these findings in a trial that actually has a priori pre-specified hypotheses around all of this stuff. So we should know in 5 years or so whether we can replicate these. But they're interesting and provocative suggestions that parenting, and especially nurturing parenting, can buffer some of the associations we see between socioeconomic disadvantage and subsequent inflammation. A positive story. We're also doing a bunch of work in pregnancy, again, trying to leverage social relationships. In that last sort of module of the talk, the focus was on parent-child relationships. In our pregnancy work, we're focusing more broadly on helping women in the middle of pregnancy build relationships that are closer either with their partners or with other pregnant women in the community. And so we're involved in a clinical trial focusing on group-based prenatal care, and having groups of 8 to 10 women go through prenatal care simultaneously, in addition to the usual medical aspects of care, to pay more attention to relationship building and stress management and coping and interactions with the medical system. This often gets called centering pregnancy. That's the formal name of the intervention. This is a riff on that. And we see in some very preliminary work that participating in this group-based prenatal care changes gene expression patterns in the placenta and in umbilical cord blood in favorable directions. These are very preliminary observational data, but they helped us to get started on a large randomized controlled trial that we're about halfway through, where we're randomly assigning 3,000 pregnant women in a small town in South Carolina to either get group-based or individual-based prenatal care, looking at birth outcomes, and then again, we're in the delivery room biopsying placentas to listen in on the gene expression patterns. So again, we'll know a lot more in a few years about how robust these associations are and how clinically valuable or not they are. All right, so good news about maternal nurturance, right? All of the data that we presented, and it's not just from us, but other groups are showing similar patterns, suggest promising indications that it's a protective factor and that it's a modifiable protective factor, and that if we make better policies and have better practices that allow for better quality family time and better quality family interactions, we might mitigate through social processes some of these socioeconomic disparities. That's my read of the data, the optimistic read. The other protective factor we've really focused on over the past few years, again, out of the developmental psychopathology literature, is self-control. And we went into this work thinking, all right, there's plenty of evidence to suggest from studies of academic outcomes and substance abuse outcomes and other mental health related outcomes that low-income kids who have high levels of self-control or high levels of executive function seem to be protected from some of the adverse outcomes that typically pattern by socioeconomic status. Thought, okay, the maternal nurturance findings transfer from psychopathology and academic outcomes to physical health. Presumably, the self-control one should too. So again, in collaboration with Jean Brody, following up on some of the samples, we started to ask these questions in longitudinal studies about whether self-control was protective against physical health outcomes for low-income kids. And the answer to this, for us, has turned out to be not so protective, actually. And this was an op-ed that we wrote in the Times some years ago reporting the early phases of this research, which suggested the troubling and somewhat paradoxical conclusion that upward economic mobility and high levels of persistence and striving might have unanticipated and unforeseen negative health outcomes, particularly for very low-income kids of color. And let me tell you a little bit about that data. This is less optimistic and cheery than the maternal nurturance findings. So I'm just going to summarize some findings here. As you may know, Jean has multiple cohorts of rural African Americans. Some of them are part of studies that started around the transition to middle school. Some of them are folks who started during the transition into adulthood. This is a study called AIM, Adults in the Making, where the participants were followed from age 17 to age 20. This is different than the intervention data that I showed you, a totally different cohort. And so what we're doing here is looking in about 300 young, late adolescent, early adults. These are all rural African Americans at psychosocial outcomes annually from age 17 to age 20 as a function of family socioeconomic status and self-control. Self-control, again, measured annually by multiple reporters, by the kid and by the parent here, and we're averaging across all those. And if these slides were bigger, what you would see here is that regardless of whether you're looking at substance use, aggressive behavior, depressive symptoms, or a more generalized composite of internalizing behavior, what predicts levels of these psychosocial outcomes from age 17 to 20 is not socioeconomic status. It's self-control, right? So you can see each of these 4 lines is basically a cell in a 2x2 matrix that through latent class analysis gives us groups that are higher low in socioeconomic status and higher low in self-control. And basically, there's a main effect of self-control. SES doesn't really matter. So again, this is very consistent with previous literature. We're not discovering anything new here. We are, at best, replicating other findings that self-control is beneficial for adolescents as they move into adulthood for many outcomes. But then if you look at physical health, and in this particular cohort, there weren't physical health measurements that were extensive. They had almost no blood. They didn't have blood pressure, but they did have some DNA sitting around because the early focus of the study was on genetics. And so we were able to harvest that DNA to look at a metric of how fast people's cells are aging. And you can do this using methylation patterns in cells to gauge basically what the person's biological age is relative to his or her chronological age. So this is an evolving metric. It's attracted a lot of interest in recent years. It's not the be-all and end-all of people's health, but it's one way when you have a limited amount of sample on health that you can start to ask questions about whether self-control relates in the same way. And what we found, to our surprise, was that actually the effects of self-control, or the relationship between self-control and the cellular aging metric, were not as favorable as they were for psychosocial outcomes, that there was this prominent interaction between socioeconomic status and self-control. And it was an interaction where if you were a rural African-American adolescent from a very disadvantaged family, having high levels of persistence and self-control was related to faster cellular aging. Your cells looked older than they should have looked given your chronological age. Whereas if you were an adolescent from one of these same rural areas and you had low levels of you came from a family that was less disadvantaged, that was working class or middle class, probably in more normative terms, that self-control was actually beneficial for you in terms of epigenetic aging, that your cells aged more slowly. And we saw this interaction not only with this particular metric of epigenetic aging, which is one that was developed by a guy named Hannum, but also with a different measure that was developed by another scholar named Hannum, and the two are correlated at about 0.2. So we see this with different measures of epigenetic aging. We also have seen this in a different sample with the same exact pattern with allostatic load. And so in two of Gene's cohorts, we see the same pattern where self-control portends better academic and mental health outcomes. And in terms of physical health, it portends better outcomes if you're a kid in an advantaged family, a relative advantaged family, and worse outcomes if you're a kid from a more disadvantaged family, that it really depends. And we really spent a couple of years scratching our heads about what the heck this means. Because it wasn't anticipated, it wasn't hypothesized. We figured that self-control would be uniformly beneficial. And in presenting these data to some colleagues of ours who came from different disciplines, who came from economics and public health and sociology, they all said, oh, of course, this makes perfect sense. What do you mean it makes perfect sense? Not in psychology. And they said, haven't you seen the data on mobility in blacks in the United States? And they showed us this graph. So this is a graph that looks at the gap in longevity between black and white Americans, right? And it stratifies it by level of education. And what you see here is that white Americans live an average of 3.1 years longer than black Americans if you just focus on Americans who don't have a high school degree, right? And if you look at high school graduates, the gap is about 3.7 years, favoring whites over blacks, right? Where's the biggest gap between whites and blacks? College degree. This isn't what you'd expect if education was necessarily the problem that needed to be solved here, right? The mortality gap between blacks and whites with college degrees is about 4.25 years, right? So the racial disparities in longevity and in health are most extreme at the high end of the education scale, right? Again, suggesting in this kind of roundabout way, in the same way our data is, that more education and potentially more affluence and maybe more striving to get that education in a country and an environment that historically has been very unsupportive is associated with some sort of physical health cost. And that's what we mean when we've talked in this editorial about hidden costs of mobility, or in some cases, some people talk about this as skin deep resilience, that this hard driving persistence that allows some Americans to live out this mythology that you pull yourself up on by the bootstraps and you work hard and you can get anywhere in America might be associated with outward indicators of success, high educational attainment, potentially good mental health, lots of income. But for some Americans, that path towards upward mobility might exact a health cost. And so I showed you a couple of different slides from South Georgia. Again, you know, in publishing these studies and thinking about these findings, we had no idea, first of all, how robust they were. We had no idea if they were something unique about rural Georgians and rural African Americans, or whether they were more generalizable to other disadvantaged groups in the United States. So we recently did some analyses of the AdHealth data set with Kathy Harris, who's the PI there, and Lauren Gaydosh, who's a sociologist now at Vanderbilt, to ask this question in a nationally representative 3-decade-long longitudinal study and get what we thought would be a more definitive answer. And so here in AdHealth, what we're looking at, first of all, the mental health outcome. This is just CESD scores, people in early adulthood. looking at it according to childhood disadvantage. So this is a index of disadvantage across family, neighborhood, and school that's done during adolescence by whether or not the subject ever gets a college degree. This is a 4-year degree. And if you look at mental health outcomes, this is depressive symptoms, you get a very clear answer, which is going to college is good for your mental health, right? Subjects who got a college degree have lower depressive symptoms between age 24 and 32. And if you redo the analyses, only focusing on people who have already finished college, you see the same thing, suggesting that this isn't necessarily a social selection effect. Point being, mobility is good for your health. It doesn't matter in this sample if you're white, if you're black, if you're Latino, or what, for the most part, your childhood socioeconomic circumstances were. If you went to college, your mental health is better. Not so with your physical health. So here, instead of depressive symptoms, we're looking at metabolic syndrome, that constellation of cardiovascular risk factors that includes abnormal cholesterol, high blood pressure, abdominal fat. What you see here is that for white Americans, going to college predicts reduced risks of metabolic syndrome, and it does so for the most part regardless of what your childhood circumstances are. But that's not true for black Americans, and it's not true for Latino Americans. For these groups, going to college is better for your long-term metabolic health if you come from a family that was middle class to advantaged in the first place. But if you come from a family that was disadvantaged, there's this apparent health cost. So again, this is a very different data set. It's much larger. We've got 3 decades of prospective data. So I think we can come to some stronger conclusions here than we could in South Georgia. None of them is a happy conclusion. They're fairly sobering. But what they do suggest is that there potentially are some costs to striving, but maybe even more importantly, there are costs to arriving at high educational success and workplace success that maybe have a lot to do with, and I can't get through this right now, our suspicion is that these are really effects about climate. That for low-income minority students who strive and succeed at the highest levels, they find themselves in academic and workplace environments that, if not aren't particularly welcoming, are in some cases outright hostile. And it's that climate that really erodes some of the physical health indices. So we have a paper that is consistent with that finding that I'll just show you here, that another postdoc, Cynthia Levine, just published in PNAS, suggesting that if you look at climates around diversity, it can tell you a lot about how the health of kids in middle school are going. And we think these school climate issues are really, really important in explaining those skin deep resilience and mobility findings. All right, I have gone too long. Thank you very much. I'm happy to take questions.